All right. So here we are talking about the urinary system. And the urinary system consists of four types of organs. There are two kidneys, two ureters, a urinary bladder, and a urethra. The kidneys are going to filter the blood and form urine. Then they will pass that urine on to the ureters, which are muscular tubes that convey that urine down to the urinary bladder. The urinary bladder will collect and store the urine until it's time to micturate or release the urine or urinate, and the urine will pass through the urethra out of the body. Now, there are two ureters, one for each kidney. Okay. The purpose of the urinary system is to filter the blood and remove metabolic wastes and toxic substances and other things that are in excess in the blood, just kind of eliminate them and restore homeostasis. If we have too much of something, we're basically going to pee it away. If you look at this diagram, you'll notice right away that the left kidney is higher than the right kidney. Okay? And here you can see again, each kidney has its own ureter. Here's one ureter, here's another, and it conveys that urine down to the urinary bladder and then out through the urethra. So the kidneys, you could see they were reddish brown and bean shaped. They have a smooth surface on the exterior, and it's enclosed in a tough, fibrous capsule. Now they lie on either side of the vertebral column in a depression on the posterior abdominal wall. They lie in the back of the abdomen, and they are retroperitoneal. Now you remember the peritoneum was the serous membrane that surrounded the abdominal organs, um, or mainly the digestive organs. Um, Retroperitoneal means that it is behind the peritoneum. All right, so the kidneys are not surrounded by peritoneum. All right. The left kidney is, again, yes, a little higher than the right kidney. And the kidney is surrounded by connective tissue known as the renal fascia, as well as masses of adipose. And when I used to teach a long time ago back in Texas, we had just anatomy as a course, not anatomy and physiology. And we spent weeks dissecting cats, and we'd go through all the organ systems. And in the most organs didn't have a lot of fat around them, but the kidneys did, and in the fat cats, they were especially fatty. The kidneys were. All right, so here you can see a cross section of the kidneys. If you look in, you can see this space called the renal hilum, and the renal hilum is just a space that allows the blood vessels to enter the kidneys as well as allows the ureter to exit the kidney. All right, good. Oh, so yes, right here surrounding the kidney, it's got that tough fibrous capsule. All right, surrounding that is a layer of adipose. As we said, the fatter the cat, the fattier the kidney was. And then surrounding that is the renal fascia. Okay, the renal fascia is a thick connective tissue layer. All right, so the kidneys, they say here, have a convex lateral surface and a concave medial surface. And what that means, if we go back here, convex means it sticks out on the side, on the lateral side, and on the medial side, it kind of caves in. It's concave. Same on the other kidney, concave on the medial surface, convex on the lateral surface. All right. Good. So the renal sinus is a hollow chamber in the medial depression of the kidney, and the hilum is the entrance to that sinus. The renal pelvis is a funnel, which is going to collect urine from the entire kidney. We also have the major calyces and the minor calyces, and this is actually kind of in reverse order. The urine flows from collecting ducts into the minor calyces, then into the major calyces, and then finally into the renal pelvis, which is again that funnel that collects all the urine and passes it on to the ureter. All right, the renal medulla is the inner region of the kidney, and it's made up of renal pyramids. They're called renal pyramids because of their uh, pyramidal or triangular shape. And then the renal cortex is the outer region of the kidney. And you may remember in the adrenal glands, we had an adrenal cortex and an adrenal medulla. So an outer layer, remember, was the cortex, and the inner layer was the medulla. Same thing in the kidney here. Outer layer is the cortex, inner layer is the medulla. And then the renal columns, these are extensions of the cortex that dip down into the medulla, and they actually give the pyramids their shape by kind of separating them. And we'll see a picture of that. And then the renal capsule we mentioned already. That's the fibrous capsule that surrounds the kidney. Now we're going to introduce a new word here, and that's nephrons. Nephrons are the functional units of the kidney. So this is the site where we actually first produce the urine, first filter the blood, and then produce the urine. And that's going to consist of a renal corpuscle and the tubule attached to it. All right. And so here you see this funnel shape. Okay, that is the renal pelvis. All right. And let's go to the other end real quick. This is the nephron, which consists again of that renal corpuscle, this round structure, and then the tubule, renal tubule, attached to it. And then at the end of the renal tubule, um, the processed filtrate is now called urine, and it's going to get passed on to a collecting duct, and that collecting duct will travel down through the renal pyramid, all right, down to the minor calyces, minor calyx singular, all right. And so here we see a minor calyx, a minor calyx, a minor calyx, and these drain down into the larger areas called the major calyces. Okay, and then the urine flows from the major calyces into the renal pelvis, which again is this funnel, and then down into the ureter, the tube that carries the urine down to the urinary bladder. Good. So, a couple other things to talk about while we're here, I guess. We've got the renal pyramids. You can see they have this triangular shape. In 3 dimensions, it would be kind of pyramidal. So, we have the renal pyramids here. And the renal medulla is made up of these pyramids. Okay. The outer layer is the renal cortex. And then the renal columns are these areas where the cortex dips down into the medulla. And you can see that the columns separate the pyramids from one another, and by separating them, it gives them their shape. All right. So yeah, the cortex is highly vascular. Okay, and so when blood flows into the kidney, most of it is going to flow to the cortex, and in the outer regions here, it's going to be processed in the renal corpuscles. Um, well, the nephrons, I should say, but that starts in the renal corpuscle. If we move back just a little bit from this view, you've got a thin section here of the cortex, and you can see that there are actually multiple nephrons running through this space that are all going to dump into the same collecting duct, and that collecting duct carries the urine down to the minor calyx. All right. So here you can see the abdominal aorta and the inferior vena cava. Okay, and so coming off of the inferior vena cava, you're going to find the renal veins. And then off of the aorta, you will find the renal arteries, okay, that supply the kidneys. All right, so for the flow of blood through the kidneys, blood passes through first the large renal artery, then into the interlobar arteries, which pass through the renal columns up to the outer cortex. In the outer cortex, you'll find the arcuate or arciform arteries. And what this name means is arc shaped, right? And so they're going to look like an arc that runs across the cortex. It'll actually run across the base of the pyramid. All right, then you'll have the cortical radiate or interlobular arteries. And the cortical radiate arteries, the name here is telling you that you find them in the cortex and they're going to radiate out, right, away from the arciform artery. Then blood passes through the afferent arteriole, which enters the nephron. Blood is filtered in the glomerulus, which is the capillary network in the nephron. And then it's going to flow out of the corpuscle through the efferent arteriole, as well as we also have peritubular capillaries, I should say. So some of the blood enters through the afferent arteriole, passes through the glomerulus, and then exits through the afferent arteriole. And then there's kind of a alternate pathway through the peritubular capillaries. We'll see a picture of that in just a little bit. From the nephron, blood returns to the renal vein through the cortical radiate veins, then down into the arcuate or arciform veins, then down into the interlobar vein, and finally the renal vein. And so you can see this is the names are the same. Basically, we're just flowing in the opposite direction. We flowed up through the renal artery to the interlobar arteries, then through the arcuate or arciform arteries, then through the cortical radiate arteries. Blood is returned through the cortical radiate veins, which drain into the arcuate or arciform veins, which drain into the interlobar vein, and then finally down into the renal vein. And so we can see a picture of that here. Here you have the renal artery, which is going to branch off and form the interlobar arteries, which run through the columns here. All right, the renal columns. Then you have the arcuate or arciform artery, which forms an arc right here. Okay, runs kind of along the base of the pyramid, but it forms an arc through the bottom of the cortex here. And then branching off of the arciform or arcuate arteries are the cortical radiate arteries. Okay, they're going to radiate out from the arciform artery. When we, let's go ahead and continue from there. Blood flows into the corpuscle through afferent arterioles. The blood gets filtered in a capillary network called the glomerulus, and then blood leaves the corpuscle through the efferent arterioles. Okay, and so eventually those they'll run their way through the cortex. We have the veins here. Okay, the veins are going to pick up fluid and certain metabolic substances and carry them down through the cortical radiate veins down into the arcuate vein, down into the interlobar vein, and then finally down into the renal vein. All right, now in the corpuscle, blood flows in through the afferent arteriole, then through the glomerulus, then through the efferent arteriole, and that will rejoin with the peritubular capillaries. The peritubular capillaries are running alongside the tubule. All right. Yeah, we've talked about this already. This is the flow of blood. You can consult it if you want, but we've talked about it. All right. right, so each kidney contains about 1 million functional units or nephrons, and each nephron is composed of two parts. You have, as we said, the renal corpuscle and the renal tubule. The corpuscle consists of a glomerulus. The glomerulus is the um, uh, filter, it's the capillary bed inside of the corpuscle. And then you have surrounding the corpuscle the glomerular or Bowman's capsule. And Bowman's capsule, it doesn't only surround the glomerulus, it's where we receive the filtrate. Basically, blood passes through the glomerulus, which is a capillary bed that filters blood, and then the filtrate, the liquid, the stuff that winds up on the other side, is dumped into the capsular space inside Bowman's capsule or the glomerular capsule. We'll see pictures of this. It'll make a little bit more sense when we have pictures. Then we have the renal tubule, which extends from the glomerular capsule to the collecting duct. And so the filtrate, it passes through the capsule and then into a region of the tubule called the proximal convoluted tubule, or PCT, down through the nephron loop, also called the loop of Henle, and then up through the ascending loop of the, well, there's a descending and an ascending loop in the loop of Henle, and then finally through the DCT, or distal convoluted tubule, and then from there, from the DCT, the filtrate flows into the collecting duct. All right, so let's look here. Here we have a corpuscle, okay, and so you can see the glomerular capsule. Okay, you can see that here. All right, it's a layer of flat cells, simple squamous here. And then here you can see the capillary bed known as the glomerulus. Okay, and if we look here at the blood vessels supplying the glomerulus, you have the afferent arteriole and the efferent arteriole. You can notice two things right away. And that's that the afferent arteriole is larger than the efferent arteriole. Okay. And blood enters the glomerulus from the afferent arteriole, flows through the glomerulus, and then exits the efferent arteriole. And uh, so what's significant about this, about the size, is that by having larger input vessel than output vessel, more fluid is going to flow into the glomerulus than is going to leave it. Okay, so the smaller size of the efferent arteriole helps maintain capillary hydrostatic pressure in the glomerulus. Okay, because there's a smaller tube leaving, that helps keep the pressure high in the glomerulus. That encourages filtration, which is the process of moving fluid and other dissolved substances, through filtration slits in the glomerulus into the capsular space. Now again, here's the capsule. So the capsular space is everything in here. Okay, all this space inside the capsule. And so just to approach this one more time, blood enters through the afferent arteriole. It gets filtered in the glomerulus. Some of the blood will remain in the glomerulus and be passed on to the efferent arteriole. Some of the fluid in the blood and dissolved substances will be filtered out of the blood. All right, so water and nutrients and wastes will be pushed through filtration slits into the capsular space. And here it's called filtrate. All right, it's not blood technically, it's filtrate that has been pushed out of the glomerulus into the capsular space. And then from here, that filtrate moves into the proximal convoluted tubule, or PCT. So, how does the filter work in the glomerulus? Well, first, the endothelial cells um, that make up the capillaries of the glomerulus are very leaky. They have gaps between them, pretty big gaps between them. Uh, they're further surrounded by podocytes, which are going to have quote unquote foot like projections called pedicels. Um, all right, and so these podocytes, they have these quote unquote foot like projections that stretch out and wrap around. And it's kind of like having another uh, mesh, like a mesh screen sitting on top of these leaky blood vessels. And the size of the gaps between the endothelial cells and the size of the gaps between the pedicels of the podocytes, uh, that basically sets up the size of the holes in our mesh screen, if we want to use an analogy. And that determines the size of the substances that can pass from the blood into the filtrate. And so some things are too big to get through this screen that we've created. All right, some things are too big to get through. Proteins generally are too big to get through. If proteins are getting through, they're going to wind up in the urine. And so if you find protein in the urine, that is probably indicative. If you find a lot of protein in the urine, that's indicative of kidney failure, specifically a condition called nephritis. But we'll talk about it in a little bit. Um, Good. I think we are good here. Remembering the names for the arterioles, by the way, afferent and efferent. So when we did the nervous system, you remember you had afferent neurons and efferent neurons, and the afferent neurons basically carried input to the central nervous system, and the efferent neurons carried output from the central nervous system to the rest of the body. And so you can think of here afferent arterioles. This is the input, efferent arteriole. That's the output. Blood enters the glomerulus from the afferent arteriole, and then it leaves the glomerulus through the efferent arteriole. All right, so uh, good. Filtrate. Once it leaves the capsule, it enters the proximal convoluted tubule. Now, why do we call this the proximal convoluted tubule? Well, just look at it. It's convoluted. It's running all over the place. And it's proximal because it's the region of the tubule that is closest to the not the glomerulus, but the corpuscle. Filtrate passes through the PCT, or proximal convoluted tubule, down into the nephron loop, which is also known as the loop of Henle. Okay. I don't normally like using the doctors' names. You know, a lot of these anatomical structures are named for doctors who discovered them, and I don't like using the doctors' names because that tells you nothing about the structure. But I will tell you, it's very common, and I'll even slip up and do it myself in the renal unit to refer to these structures by their doctor's name, right? So Bowman's capsule, you'll probably hear that a lot more often than you hear glomerular capsule. And loop of Henle, you'll probably hear that a lot more often than you hear nephron loop. All right, so yes, here we have the loop of Henle. You have the descending limb, and then you have the ascending limb. And most of the descending limb is going to be thin, and then the ascending limb is going to have a thin and a thick portion. All right, so filtrate passes from the ascending limb of the loop of Henle onto the DCT, or distal convoluted tubule. and then from the distal convoluted tubule onto the collecting duct, which passes the urine. It's urine now. Urine onto uh, the minor calyx, the major calyx, the renal pelvis, the ureter, the urinary bladder, and then the urethra. All right, so again, why is this the proximal convoluted tubule? Why is this the distal? You can even see usually the distal is in close contact with the capsule. This one's proximal because it's the first region you come to, the first region of the tubule. The distal convoluted tubule, even though it may be closer to the capsule, is further down the line in the tubule, and so we use that term, distal convoluted tubule. All right. Good, good, good. So you can see here we've got the blood entering through the afferent arteriole, passes through the glomerulus, gets filtered. Some of the filtrate winds up, some of it winds up as filtrate in the capsular space, passed on to the PCT, loop of Henle, onto the DCT, and then finally uh, the collecting duct. Now, the blood that leaves the glomerulus enters over here and it splits and it goes to the peritubular capillaries, which run along the tubules. And then we have this structure down here called the vasa recta, which is kind of an alternative path for the blood. And here in the vasa recta, you can see that it closely follows the nephron loop. Okay, closely follows the nephron loop. And we'll later see some sort of countercurrent exchange, it's called, where we're exchanging nutrients between the blood and the vasa recta and the peritubular space, basically the interstitial fluid here, which is going to allow us to excrete and reabsorb certain allow us to secrete and reabsorb certain substances. All right, so moving on. Here you can see a glomerulus under the microscope. You can see that capsule surrounding it, and then a little bit of that capsular space in here. All right, it's a little hard to make out. You can see a clear tubule here, right? You can see that clear shape. Otherwise, a little difficult to make out. All right, so we've talked about this, the flow of filtrate as it becomes urine, and then all the way out of the body here. All right, so you have two types of nephrons. You have the cortical nephrons and the juxtamedullary nephrons. And juxtamedullary means that they are next to the medulla. Cortical nephrons mean, of course, they're going to be found in the cortex. And so, although the corpuscle for all of these nephrons will be found in the cortex, cortical nephrons typically have a corpuscle that's much higher in the cortex, and then their loop of Henle, it might dip down into the medulla, but it doesn't go very far. The juxtamedullary nephrons have a corpuscle that's much lower in the cortex, and it's going to be close to the medulla, and then its loop of Henle will be much longer and it'll dip much further into the renal pyramids. And so, although the majority of nephrons are going to be cortical nephrons, we're going to see a lot of the filtration occurs in the juxtamedullary nephrons. They're important for regulating water balance. Good. We also have a juxtaglomerular apparatus. That is a difficult word, juxtaglomerular. There's an art term you may have heard, juxtaposition, where you put two things next to each other and then their being next to each other makes you think about them in a different way, right? If you've never heard that term, don't worry about it, but if you have, it might help you remember juxta means next to. So the juxtaglomerular apparatus involves um, cells in the distal convoluted tubule as well as cells surrounding the capsular space there, which are going to detect ion concentration as well as blood pressure. So you have a region called the macula densa or dense spot, which has tall, closely packed cells found in the ascending limb. And then you have juxtaglomerular cells nearby, which are large, vascular, smooth muscle cells found in the afferent arteriole. So here you can see those juxtaglomerular cells kind of between the afferent and the efferent arteriole, also between those arterioles and the ascending limb of the loop of Henle. The macula densa is right up against the juxtaglomerular cells. That's the dense spot. We've got these tall cells right here. Okay. Um, and so they're going to detect blood pressure, basically also salt content. And salt, of course, is closely related to blood pressure, right? Um, and so if blood pressure drops, they will release renin. And then renin is going to activate a precursor molecule called angiotensinogen. Remember that O-gen means it's a precursor molecule, precursor inactive protein. So renin is an enzyme that activates angiotensinogen. And when it does, it turns it into the protein angiotensin 1. Angiotensin 1 passes through the bloodstream, and it's going to reach the lungs where we have an enzyme called angiotensin converting enzyme, or ACE, or ACE. And ACE is going to convert angiotensin 1 into angiotensin 2, and then angiotensin 2 will go on to have a variety of effects on blood pressure. It will raise blood pressure. And so if your blood pressure is already high, your doctor may prescribe you an ACE inhibitor, and that's a drug that will inhibit angiotensin converting enzyme and basically prevent you from producing angiotensin 2. Um, but anyway, it all starts down here in the kidney. All right, we're detecting the blood pressure in the kidney, and when blood pressure is low, we will release renin, which starts the activation of the renin angiotensin system. All right. So nephritis is inflammation of the kidney. You may have noticed already that renal refers to the kidney. Nephro also refers to kidney. I believe renal is probably Latin. Nephro is probably Greek. Um, anyway, nephritis is inflammation of the kidney. Glomerulonephritis is inflammation of the glomeruli, and it can be acute or chronic. In acute glomerulonephritis, we're going to see it results from an abnormal immune reaction about 1 to 3 weeks after infection by streptococcus. And the infection does not start in the kidney, but antigen antibody complexes move through the blood and they kind of block the glomerulus. You remember the glomerulus is not just a capillary bed, it acts as a filter for the blood. And these antigen antibody complexes will kind of lodge in the filter slits there and kind of block him. So the complexes get deposited in and, yes, block the glomeruli. In chronic glomerulonephritis, we see this as a progressive disease where more and more nephrons get damaged over time until the kidneys cannot function. You'll see prolonged inflammation in this case, and then fibrous tissue is going to replace the glomerular membranes, basically the capsules, and then disable the nephrons. Not the capsules, sorry, the endothelium of the glomerulus, making them non-functional. All right, the main function of the nephrons and the collecting ducts is to control the composition of the body fluids and remove waste from the blood. The product of this process is urine. Basically, again, we are maintaining homeostasis by controlling what is in the blood. Now, the liver does some of that also. The liver's just full of enzymes, all kinds of enzymes. They're going to do all kinds of things for converting one substance into another, that kind of thing. But when we have too much of a substance and we need to get rid of it, we're going to get rid of it in the urine. All right. So urine contains waste, excess water, and electrolytes. So the three processes of urine formation are glomerular filtration, which we've talked about a little bit already, tubular reabsorption, and tubular secretion. Okay. Glomerular filtration is where we first filter the blood, all right, and we produce that filtrate in the capsule. Tubular reabsorption is where this filtrate passes through the tubules, and then we are going to reabsorb into the interstitial space of the kidney some of the substances that made it through that we don't maybe don't want to pass on, that we maybe don't want to get rid of. And then tubular secretion is going to involve the movement of substances from the vasa recta and the peritubular capillaries into the tubule. Basically, not enough of a substance got filtered, or maybe it was too big to get filtered, and we're going to move it into the kidneys through a process called secretion. Sorry, we're going to move it into the urine through a process called secretion. All right, so up top you've got a normal capillary bed. This is a diagram of a normal capillary bed. You've got an arteriolar end and a venular end, and then in between are the capillaries where we exchange oxygen and nutrients and things like that with the interstitial fluid, pick up waste, and return that through venous blood flow. We all know this. Down here's the one that's new to us. We've got the glomerulus here with the afferent arteriole, the capillary bed known as the glomerulus, and then the efferent arteriole. And then the blood will flow through the peritubular capillaries, and there we will exchange oxygen and other nutrients with the interstitial fluid, the tissue fluid, and we'll pick up certain metabolic wastes and carry them on down through the venular end. Now, filtration occurs here in the capsule where water and dissolved substances are pushed out into the capsular space. They're filtered out, and so this is filtration. That filtrate moves through the tubule. Reabsorption is where maybe there's something in the filtrate that we don't want to get rid of. We're going to reabsorb it into the peritubular capillaries and return it to the bloodstream. And then certain substances that we have too much of in the blood, we may, and maybe didn't get filtered out in the glomerulus, we're going to secrete that substance and pass it on to the tubule to make sure it winds up as urine. All right, so we've talked about glomerular filtration. Let's see, is there anything here we missed? Water and small dissolved molecules and ions, they get filtered. Large molecules like proteins, again, don't get filtered. They're too big to fit through the filtration slits there. Most proteins are too big. The main proteins in your blood plasma are albumins, and they are way too big to fit through. Under normal circumstances, we would say under physiologic conditions, they're too big to pass through. So the glomerular capillaries, they're way more permeable than other capillaries due to fenestrae. That's a term I didn't use earlier. It comes from the Latin meaning windows. So these are basically tiny openings in the walls of the endothelium, big gaps between the endothelial cells. Those are the fenestrae, or just windows. Right? The glomerular filtrate is formed as the substances are filtered from the glomerulus into the glomerular capsule. We've covered that. All right. So filtrate has about the same composition as tissue fluid. It also has about the same composition as, well, as blood, just minus the large proteins, right? And tissue fluid's the same roughly as blood also, just minus the tissue, not tissue, minus the plasma proteins, which are too large to enter the tissue. All right, so we've seen glomerular filtration. We've talked about this plenty at this point. Here you can see some of those fenestrae right here, those openings. Again, literally Latin for windows, openings through the endothelium, which allow fluid and small substances through. This is further surrounded by the podocytes, which have their foot-like extensions called the pedicels, right? And so these two things together form a kind of a mesh network that allows, uh, yeah, water and small substances to pass through. All right, so the main force that moves substances by filtration through the glomerular capillary wall is hydrostatic pressure. And this is the hydrostatic pressure of the blood inside the glomerulus. The afferent arteriole has a larger diameter. It's bigger, as we said, than the efferent arteriole. And so this creates resistance in the efferent arteriole. And as the resistance there increases the blood pressure in the glomerulus, we're going to see an increase in filtration. So the net filtration pressure is the force favoring filtration minus the forces opposing filtration. And so what would favor filtration? That would be high blood pressure, um, right? High blood pressure. Forces opposing filtration would be a high oncotic or osmotic pressure in the blood. All right. Um, Good. Also, capsular hydrostatic pressure, right? So let's go back and let's look at a picture and talk about these, I guess. All right, so what's going to favor filtration? That's high hydrostatic pressure. That's the literal blood pressure, the pressure of the fluid inside the glomerulus. If that hydrostatic pressure is high, that's going to push fluid out of the blood and into the capsular space. Now, what forces oppose this? We've got the colloid osmotic or oncotic pressure, and that's the pressure of the blood. Um, no, that's related to the concentration of solutes in the blood. Okay, and so if there's a lot of, say, protein in the blood, that's going to attract water out of the capsular space and back into the blood. And then you've also got the capsular hydrostatic pressure. And so the pressure in the glomerulus, of the blood in the glomerulus, that blood is under pressure. But the filtrate out here in the capsule is also under pressure because this area is filled with fluid, and this fluid, um, it can only go one place, and that's out through the proximal convoluted tubule. If you have a blockage uh, down the way, down the tubule, um, or a kidney stone, for instance, we'll talk about those a little bit later, but if you have a kidney stone, that's going to block the flow of urine from the PCT through the loop of Henle, through the DCT, through the collecting duct, into the minor calyx, major calyx, etc. So all that fluid is going to back up, and if you have that blockage, that fluid, that urine, that filtrate, it backs up into the capsular space. And then you actually won't have a flow of fluid through the glomerulus into the capsular space. You would have, um, you'd have that fluid back up. You would not be able to filter the blood anymore. Again, luckily we have two kidneys, so if you have one blocked, the other can take over. But All right, so those are the forces favoring and opposing filtration. Even if you don't have a kidney stone, by the way, I should just say this. Even if you don't have a kidney stone, there is fluid in here and it is under pressure. And so even though it's at a lower pressure than the hydrostatic pressure of the glomerulus, it will oppose filtration. All right. Now, not totally, again, because the hydrostatic pressure is higher under physiologic conditions, you will have net filtration, right? You will still have some flow, a net flow of fluid from the glomerulus into the capsular space under physiologic conditions. All right, so we kind of talked about these things, but we can go ahead and put some numbers to them. All right, so here we have the capillary. Okay, you see the blood flows through, and the glomerular hydrostatic pressure is a large force here, and it's been measured at 60 millimeters mercury. Okay, now the colloid osmotic pressure or colloid oncotic pressure, whichever term you like, is the pressure, the osmotic pressure generated by just the presence of solutes. And so in this case, it's going to be large plasma proteins. They attract water, they draw it in. And so the pressure's been measured at negative 32 millimeters mercury. In other words, the force is inward because it's negative. All right. And then next up, you have the capsular hydrostatic pressure, the pressure of the fluid also already in the capsular space, and that's been measured at negative 18 millimeters mercury. And so if you combine these numbers, you get 60 minus 32 minus 18, leaves us with a net filtration pressure of 10 millimeters mercury. That is the net filtration pressure at under physiologic conditions. Basically, normally, that's the net filtration pressure. Now, what happens if we change these numbers? Let's say you have a high protein concentration. That's going to increase the colloid osmotic pressure. And so we could increase that to a number like 40 millimeters mercury, which would change the net filtration pressure to 2 millimeters mercury, right? Or we could, and in that case, you'd see a decrease in filtration, which would be bad. We need filtration to occur. We need to filter the blood. Uh, we mentioned the kidney stones also. If you have a blockage that is going to increase the capsular hydrostatic pressure, that can also decrease the net filtration pressure. And if this number reaches zero or even becomes negative, you're in trouble. All right, so the glomerular filtration rate is proportional to the net filtration pressure. The average adult has a GFR or glomerular filtration rate of about 125 milliliters per minute, and that's about 180 liters per day. Um, so the total blood plasma gets filtered about 60 times per day, but only a small percentage of the filtrate actually gets excreted as urine. Now, this is a big number. You may think to yourself, uh, so I filter through the kidneys 180 liters of fluid. 180 liters gets turned into filtrate every day. I don't pee 180 liters a day, right? In fact, most people, it's probably around 500 milliliters to 1 liter. All right. In fact, the number they're giving us over here is about half, zero point 0.6 liters to 2.5 liters, depending on what you're drinking, how much of it you're drinking. All right. Now, there's a great disparity between these numbers, and the reason for that is that the vast majority of the fluid that gets filtered is going to get reabsorbed. We are going to reabsorb the vast majority of the water that winds up in the filtrate. In fact, our goal normally is to produce the most concentrated urine that we can. All right. Yeah. Unless we're overhydrated, some people overhydrate, and then you do need to get rid of the excess water. Under physiologic conditions, I should say, our goal is to make the most concentrated urine we can. So the glomerular filtration rate is going to remain pretty constant, and this is through a process called autoregulation. So certain conditions are going to override the autoregulation, like when the GFR increases in the case of excess body fluid. So mechanisms that keep GFR constant, we mentioned autoregulation, as well as increased sympathetic impulses. They're going to keep the GFR constant under changing blood pressure conditions. And they do this by affecting the afferent and efferent arterioles, causing them to constrict simultaneously. Then we have the renin-angiotensin system, which is going to keep blood pressure high. Well, it's going to raise blood pressure if blood pressure has dropped. And then we have other hormones like the atrial natriuretic peptide, ANP, as well as the ventricular natriuretic peptide. Now you may notice this is BNP. Why B and not V for ventricular? And that's because it was originally thought that this peptide came from the brain. It actually comes from the ventricle. All right. Now, so the renin-angiotensin system, we mentioned earlier, the liver produces angiotensinogen, the inactive precursor. When blood pressure drops, the kidney, through the juxtaglomerular apparatus, will detect that drop in blood pressure, and it will release renin, which activates angiotensinogen to form angiotensin 1. Angiotensin 1 circulates the bloodstream and then it reaches the lungs. And in the lungs, that's where you find the enzyme, angiotensin converting enzyme, or ACE. And it's going to activate angiotensin 1 to form angiotensin 2. Then angiotensin 2 has a variety of effects. It'll cause vasoconstriction. Constricting the blood vessels increases blood pressure. You're going to see increased aldosterone secretion. Remember, aldosterone causes us to retain sodium, basically reabsorb it from the urine. Uh, and reabsorbing sodium causes us to also reabsorb more water. Um, and so that'll increase blood pressure by increasing blood volume. And then angiotensin 2 is also responsible for increasing antidiuretic hormone secretion. Antidiuretic hormone is another hormone that affects the kidneys, and we're going to see that we reabsorb water in the distal convoluted tubule. And then you'll see increased thirst, so you'll go out and drink more to, again, raise the blood volume, which raises the blood pressure. All right, so tubular reabsorption. Actually, I think I'm going to end it here tonight.